Hello, eighth graders. Today we're going to be talking about Punnett squares. This may be the first time you've seen Punnett squares. You may have seen these in the past, but basically a Punnett square is a graphical representation of the possible outcomes of an offspring that's the result of a particular breeding event. So in order to create a Punnett square, you do need to have information about the genotypes or the genetic makeup of both parents. So here you can see we've got parent number one. And here, parent number one is called homozygous dominant, which we'll talk about in a second. And here we've got parent number two. So parent number two in this cross is going to be homozygous dominant as well. So this is one of the simpler crosses that you can look at. So I wanted to bring you over here to this little section that kind of defines all the different things that you need to know about filling in a Punnett square. So the first thing is, what is a genotype? So the genotype is the combination of alleles for a particular gene. So here you can see we've got our alleles, and the genotype for parent one is big B, also known as homozygous dominant. So the next thing is, what is the phenotype? The phenotype of an organism is the physical appearance of the genotype. So the physical appearance would be something like the color, the shape, or the size of an organism. In this example, we're crossing two little guys here, and in this cross, blue skin, so I'm going to just underline that, blue skin is dominant to purple skin, okay? So what this is telling us is that blue skin is dominant, so it's going to be big B, and purple skin is going to be little b. So fill in the information about each parent based on the information given. So we'll do that in just a second. So coming back over here, the phenotype, our phenotypes in this cross will either be blue skin or purple skin. Excuse me, depending on which genotype we have. Dominant. Dominant alleles are alleles that show their effect even if only one is present, okay? They show their effect even if only one is present. So what's an allele? An allele is just a version of a gene, okay? So here we have the gene for skin color. So blue skin is dominant. That's the dominant allele. Purple skin is recessive. That's the recessive allele. So we've got two different alleles. If you're said to be recessive, then that's the allele that can only show its effect if it's paired with another identical recessive allele, which will make more sense in a minute when we start doing these, crosses. Homozygous is when the alleles for the trait are the same. Remember that prefix there means same, okay? And we talked about that with chemistry earlier on when we talked about mixtures and homogeneous mixtures, everything looks the same. Heterogeneous mixtures, you can see the different parts. So in homozygous alleles, that's when the alleles for the trait are the same, like big R, or little r, little r. And in heterozygous, that's when the alleles for the trait are going to be different. So big R, little r is heterozygous. So how do you fill in a Punnett square? Well, I like to start with the left and bring my left letter over and then bring my top letter down. So here, if I bring my big B over and big B down, big B over and big B down, big B over and big B down, okay? So to calculate the percentages here, we're just gonna look at, we have 4 boxes, and 4 out of 4 boxes are big B, okay? So each box represents 25%. So what are the possible genotypes from this cross? We only got big B. What are the possible phenotypes from this cross? Well, if we go back here, we see in this cross, blue skin is dominant. Since we had all dominant, we only could have blue skin. What percent would be purple? Well, you would need 2 recessive alleles to have any purple organisms, and we didn't have any. So we would actually have 0% purple because we had no little b, little b. What percent would be blue? 100%, because all of them had at least one dominant allele. And what percent would be heterozygous? So let's just go back here and see. That's when the alleles for a trait are different, like big R, little r. We didn't have any of that, so 0% were heterozygous. Okay? Just going to move on to the next example. So all of this stuff is the same. Graphical, a Punnett square is a graphical representation of the possible outcomes, and we still have all of our information, which you're going to want to write down so that when you start working on some of these questions, you can refer to this to help you, okay? So here we're crossing, parent 1 is homozygous dominant, so they're big B, big B. And parent number 2 is heterozygous dominant, so that person is big B, little b, because remember, hetero means 2 different, so they have 2 different alleles. So it's going to look a little different this time when we fill in our Punnett square. So I'm going to bring my little b over here and my big B down. You'll notice that I drew the little b second. I always put the lowercase letter second and the capital letter first. So I'm just going to keep doing that. So little b over, big B down. Here we've got big B, and big B, big B. And remember, each of these boxes represents 25%. So what are the possible genotypes from this cross? My genotypes would be big B, little b, and big B, big B. So what are the possible phenotypes from this cross? Well, every single option had a dominant allele. So I go back and I remember, that's right, blue is dominant, purple is recessive. So I only could have blue skin. What percent would be purple? Well, 0%, because the only way to express a recessive allele is to have 2 of them, and I only have 1. So 0% would be purple, 100% would be blue. Here I have 50% heterozygous, big B, little b, big B, little b. Okay, you see that? So I'm going to add 25 and 25 gives me 50%. Move on to the next example. So here I've got parent 1 is parent 1 is heterozygous dominant, so big B, little b. And parent 2 is also heterozygous dominant, big B, little b. So we say dominant because they're going to express the dominant allele. That's why it's called dominant. So again, blue skin is big B, purple skin is little b. All right. So I'm just going to bring my little b over and my big B down. Little b over and little b down. Big B, big B, big B, and I always draw the lowercase second, little b. So each of these boxes, again, represents 25% of the whole picture. So when I answer my questions, what are the possible genotypes from this cross? I have big B, little b, little b, little b, and big B, big B. What are the possible phenotypes from this cross? I actually can have purple skin, or all 3 of these all have a dominant allele, and so they're all going to be blue skin. So I can have purple or blue. What percent would be purple? Only 25%, because only 1 box out of 4 is homozygous recessive. So 25%. What percent would be blue? 3 out of 4 boxes are going to be blue. And so 75% would actually be blue for this example. And then what percent would be heterozygous? Heterozygous is when the alleles for a trait are different. So 1, 2. So I have 25 plus 25 is 50%. Okay? What I would like for you guys to do at home is to take the last cross, and I'm just going to actually look at it. The last cross is homozygous dominant and homozygous recessive. I'd like